Okay, in this screencast we're going to have a bit of a look at protein production. It's going to involve us understanding a little bit about the structure of proteins, but also using that with the structure and function of the organelles of a cell. We'll have a bit of an overview of the processes of transcription and translation, and these are both things that we're going to look in a lot more detail when we study DNA in Unit 4. So let's start with an overview of protein structure. And first we know that we start with the primary structure, just putting the amino acids in order along the molecule. Next is the secondary structure, where we use the hydrogen bonds in between the strands to make the alpha helices and the beta pleated sheets. In the tertiary structure, that's where we get that overall shape of the polypeptide and get that individual shape that's so specific and get that molecular specificity. Finally, it's a matter of getting a couple of these polypeptides and putting a few of these chains together to make the final product, and that's a quaternary structure. So let's have a look at the organelles involved. First off, we have the nucleus, which is this overall structure. This has all the DNA, which are the instructions. They get changed to messenger RNA, which comes out through these small pores to the rest of the cell. Inside the nucleus is also the nucleolus, and that is basically the organelle responsible or has the instructions for making the ribosomes, which we'll talk about later. Here's an electron micrograph just showing this structure. Next we have the endoplasmic reticulum. This is where the construction of the proteins actually happens, and it happens on these small structures along this folded membrane called the ribosomes. Okay, and we can often see, like electron micrographs, this folded structure often around the nucleus with these small dots, which are the ribosomes. The final organelle we'll consider here is the Golgi apparatus or the Golgi body, and this is where that final construction, where different parts of the molecule are put together and some of these polypeptides are constructed into a product. We can see these blobby little bits that sometimes appear off the Golgi apparatus, and this is basically where vesicles or little bits of membrane form around the product and they exit the cell by exocytosis. So just looking at this in an overall diagram of a cell, we start off in the nucleus with the DNA, which has the instructions. They get changed to RNA, which come out through the nuclear pores to the endoplasmic reticulum and the ribosomes. That's where the protein construction occurs. The nucleolus inside the nucleus is responsible for the construction of these ribosomes, which appear on the endoplasmic reticulum. And our final organelle involved is the Golgi body. The polypeptides that are made at the endoplasmic reticulum could be transported to the Golgi body and then secreted out through the cell. So let's just briefly look at two of the terms that we're going to use when we talk about the synthesis of polypeptides or the making of polypeptides. One's transcription and one's translation, and we'll look at these in a lot more detail in Unit 4. But for now, transcription is copying the code from DNA to RNA, changing from one type of code to another. And that's making the messenger RNA molecule, which travels out of the nucleus to the endoplasmic reticulum. Okay, really you couldn't have the DNA going out to do it, so we need a messenger molecule to go out and do it. The next stage is translation, and translation is making the polypeptide from the messenger RNA. And that's the actual assembly of all the amino acids in the correct order to make that particular molecule. When this occurs, we have three types of RNA molecules involved. The first is a messenger RNA, and that is the instructions that comes as a result of transcription. The ribosomal RNA, which is the RNA that makes up those ribosomes, and that's just the site where it happens really. And the transfer RNA, and that has an interesting job. That actually transports the amino acids to the correct spot on the messenger RNA at the ribosome. So it's just taking the right amino acids across so the molecule can be constructed. So let's use this diagram here to have a look at these processes of transcription and translation and sort of piece everything together. So we're basically inside a cell, and this is the nuclear membrane here. So this whole structure around here would be the nucleus. And what we have here is we have two things happening. We have the DNA, which is transcribing some instructions to messenger RNA. The DNA obviously can't travel outside the nucleus to construct proteins, so it sends a special messenger molecule, this messenger RNA, and this travels out of the nucleus through this nuclear pore. The other thing that comes from the nucleus via the nucleolus is the ribosomal RNA, and this travels to the endoplasmic reticulum to construct the ribosomes. The other process we have is translation, and this is actually making this continuous chain of polypeptides at the ribosome. And how this happens is the messenger RNA has instructions, the order of the amino acids need to appear, and it has these instructions in little triplet codes called codons. The transfer RNA takes these amino acids to the correct spot on the messenger RNA, and we gradually build this continuous chain of amino acids, making a polypeptide chain. I hope this has helped with the understanding of protein synthesis, and good luck.